Welcome to the Proteomics course. Today we will talk about genomics and transcriptomics, and then we talk about why to study proteomics. So, lecture outline we will talk about genomics, transcriptomics and need to study the proteome by using proteomics. Understanding biological system is very challenging task. During the last decade, we have witnessed the revolution in biological research as this discipline has fully embraced omic technologies. The emergence of genome-wide analysis to understand DNA, RNA and protein by employing genomics, transcriptomics and proteomics has revolutionized the study of control networks that mediate cellular homeostasis. So, let us talk about genomics. It has been only 60 years since the 1953 landmark study of Watson and Crick deduced the double helix structure of DNA, and biological science research has witnessed a great progress in genomic research during the last decade. The simultaneous effort of Human Genome Project and Cellular Genomics completed the sequencing of human genome in 2001. So, let us look at some of the definitions. Genome, it is the entire sequence of an organism's hereditary information, including both coding and non-coding regions encoded in DNA, which is known as genome. Studying genome of an organism by employing sequencing and genome mapping techniques is known as genomics. The genome sequencing projects produced massive amount of information on DNA sequences for many species. The genomics research is also providing information for mutations, deletions and epigenetic alterations which modulate gene expression. The various sequencing methods have been employed for genome sequencing. Let us first discuss the traditional methods. The traditional DNA sequencing was done by using Sanger's method. As you can see in this image here, the genomic DNA is fragmented, cloned into a plasmid vector and used to transform E. coli. The fluorescently labeled dideoxynucleotides or DDNTPs terminate DNA extension reaction. In a single tube, 4 labeled DDNTPs are added, and in a capillary base electrophoretic gel, the colored DNA fragments are separated. Then a laser beam is applied which detects color and determines the sequences. A simple and elegant method for DNA sequencing was devised by Frederick Sanger, where a collection of DNA fragments are synthesized by means of controlled interruption of enzymatic replication. 4 DNA synthesis reactions are carried out simultaneously, with the strand whose sequence is to be determined being used as the template. The reaction mixture consists of regular deoxynucleotides and DNA polymerase along with a small amount of one labeled dideoxynucleotide analog being added to each of the 4 reaction mixture. A primer is added to begin the DNA synthesis and strand elongation continues until a dideoxy analog gets added instead of the regular DNTP. The chain termination occurs at this stage due to the absence of a 3' hydroxyl group for the formation of next phosphodiester bond. The synthesized strands are separated from each other, after which the differentially labeled strands of various length are separated by electrophoresis. The smallest fragment move further in the gel, while the larger fragments remain close to the point of application. The different fluorescent labels of each DDNTP can then be detected by scanning the gel with a beam of laser. The output sequence obtained is complementary to the template strand, which can be used to deduce the original desired template sequence. To obtain the human genome sequence, the Human Genome Project employed a strategy of shotgun sequencing. So, from a genomic library, clones were isolated and ordered into a detailed physical map. Further, the individual clones were sequenced by shotgun sequencing. The Human Genome Project group produced a working draft of human genome by a map based strategy, while Celera Genomics sequenced the human genome by whole genome shotgun method. Genomic DNA is cleaved using a suitable restriction endonuclease and the fragments inserted into bacterial artificial chromosomal vectors. These vectors enable the DNA fragments to be amplified. The genomic DNA fragments of the library are then organized into a physical map, after which individual clones are selected for sequencing. The selected back is amplified and these clones are sequenced using Sanger's chain termination method. The sequence of the clone is then deduced by aligning them based on their overlapping regions. The entire genomic sequence is then obtained once each back is sequenced in this manner. Let us compare some of the traditional DNA sequencing methods. As you can see here, the chain termination method or Sanger method, that is a gold standard, but it is very time consuming. Sensitivity is high, but in pyro sequencing, which is based on Kb luminescent detection system, the sensitivity is very high. Even multi-TOF can also be employed if your aim is to identify variations in SNPs and sensitivity is very high. So, let us discuss about the genome sequencing projects. The genome sequencing projects aim to decipher the complete genome sequence of all chromosomes of an organism. Several such projects were initiated and successfully accomplished. Sequencing the genome of higher organisms such as human was very challenging, but researchers have shown very good team effort and successfully completed the draft sequence in 2001 and complete sequence in 2003. Here on the time scale, you can see the sequencing of various organisms which was progressed from 1990 to 2001. The Human Genome Project provided insights for various details, such as the total number of genes are estimated to be around 25,000. The average gene consists of 3,000 base pairs. The human genome consists of 3.2 million base pairs. Human genome sequence is exactly the same, almost 99% in all the people, and about 2% of the genome encode instruction for the synthesis of proteins. The various potential benefit of Human Genome Project. The information was useful for molecular medicine, environment, agriculture and livestock, microbial genetics, risk assessment of individuals for specific allergens, as well as in other fields such as anthropology, evolution and human migration. Several genome sequencing projects that aim to elucidate the complete genome sequence of organism have been undertaken by several research groups all over the world. The DNA sequences are identified by the shotgun sequencing technique and then aligned using suitable software to provide the complete genome sequence. The genome sequence of a large number of prokaryotic and eukaryotic organisms have been successfully deduced. The immense amount of information held by the human genome motivated researchers to understand the nature and content of genetic material in great detail. A collaborative effort between 6 countries and 20 laboratories was undertaken in 1990 to produce a draft of human genome sequence. Work proceeded rapidly, with a draft covering most of the genome being completed by 2000 and a greater coverage being achieved by 2003. Before sequencing the entire genome, physical maps of the chromosomes were made. This helped to provide key tools for identification of disease genes and anchoring points in genome sequence. Pilot projects were then launched to create a draft of the genome sequence. The shotgun approach was fundamental technique which was used for large scale sequencing of human genome, which also make use of Sanger sequencing. The collaborative effort to sequence the entire genome was challenged in 1998 by a privately funded organization which aimed to finish the sequencing before the publicly funded group. Progress made in sequencing was very rapid, and by 2001 a draft of the sequence was ready, covering around 83% of the genome. After successfully sequencing the human genome, the technology of sequencing got advanced, and now there are next generation sequencing methods are available. So, human genome sequence project was completed in almost 12 years at a cost of around $3 billion US. Very recently, the next generation sequencing strategies have dramatically increased the pace of sequencing by several order of magnitudes and also reduced the per base cost of raw sequence significantly. The individual genome sequencing, metagenomic studies and SNPs and mutant analysis is also possible due to the next generation sequencing methods. So, traditional sequencing, which we discussed earlier, including the Sanger's method of chain termination method as well as chemical method Maxim and Gilbert in 1976, those can be termed as the first generation sequencing methods. The next generation or second generation sequencing technology, it includes sequencing by ligation or by synthesis, including the pyrosequencing and reversible chain termination. Even more advanced form, the third generation sequencing technology is also in progress, which is aiming to further improve the second generation sequencing technology and lowering the cost per base by using scanning tunnel electron microscope, fluorescence resonance energy transfer, single molecule detection and protein nanopores. The next generation sequencing based on nanopore structures is known as nanopore sequencing, which is shown in this slide. This is a single molecule DNA sequencing technology that does not require any fluorescent labeling and makes use of any changes in electrical current produced when single nucleotides pass through the nanopores. The individual base detection is achieved through the measurement of conductivity either across or through a membrane via a nanoscale pore. This innovative method offers a label-free approach for DNA sequencing. An exonuclease cleaves the single-stranded DNA one base at a time to release the nucleoside monophosphates. These NMPs pass through the nanopore under an applied potential which is covalently coupled to an adapter molecule. The continuous movement of NMPs through the nanopore results in characteristic fluctuation of electric current, which enables detection of various nucleotide bases. Multiple rounds of nucleotide addition are carried out on the immobilized template DNA by using DNA polymerase in the presence of ATP sulfurylase, luciferase and the nucleotide degrading enzyme apyrase. The release of an equal amount of pyrophosphate is determined by its conversion to ATP by ATP sulfurylase, which in turn is determined by the release of light on reaction with luciferase. The amount of light produced is determined by means of a CCD camera, which is used to determine the nucleotide added and therefore, the sequence of the template DNA. The various next generation sequencing commercial platforms currently available will very briefly talk about the pros and cons of each of these methods. The Illumina method, which is a flow cell based method, is employing reversible dye termination and uses 4-color optical imaging. It is used for whole genome sequencing and very widely used, but one of the limitation is low multiplexing capability. The pyro sequencing or 454 pyro sequencing is based on bead based method with emulsion PCR and CCD light imaging. It is used for targeted exon sequencing. However, the contamination risk due to the emulsion PCR is one of the limitations. Helicos. Here oligo DT captured poly A tailed DNA fragments and optical imaging is used. The method is used for whole genome and single molecule sequencing. High NTP incorporation error is one of the limitations. Supported oligonucleotide ligation and detection, known as SOLID, is based on sequential dinucleotide ligation and optical imaging. It is used for whole genome sequencing and SNP detection. Limitation is the longer run time. Last method is ion torrent method, which is a standard DNA polymerase chemistry based method, where semiconductor based non-optical detection method is used. It is used for targeted sequencing. These are only few representative platforms which are described here, but there are many exciting commercial platforms are also available currently for next generation sequencing. So, let us discuss the advantage and disadvantage of next generation sequencing versus the traditional Sanger based sequencing. In next generation sequencing, the preparations are done in vitro, whereas in Sanger's method, as we discussed, the transformation of E. coli is done. So, these type of limitations can be avoided by using NGS method. The NGS method is based on arrays, not the capillary gel based method, which is employed in Sanger's method. So, sequencing time is reduced in NGS method. The cost is reduced in NGS method because all the polonies can be treated with single reaction volume, which reduces the overall cost. The next generation sequencing methods can detect deletion, translocation, copy number alterations, etc., with very high frequency. However, very sophisticated bioinformatic tools are required for data analysis of next generation sequencing, and data interpretation is still remain challenging. So, some of these are still limitation of next generation sequencing platforms. Now, let us talk about another interesting technology commonly employed in genomics as well as in transcriptomics, the DNA microarrays. The DNA microarrays can be prepared from any DNA sequence by chemical synthesis or polymerase chain reaction. The DNA is printed on the glass slide or other substrates. DNA can be cross-linked by UV light or employing other immobilization chemistries. Once ready, these microarray slides can be used for various applications. So, let us talk about a typical microarray experiment where aim is to compare an healthy sample with a disease treated sample. So, from both the populations, the mRNA has to be extracted, then mRNA should be converted to cDNA by enzyme reverse transcriptase, as you can see the second step here in the slide. During this step, label samples using the fluorescently labeled deoxyribonucleotide triphosphate. On a microarray slide, incubate labeled cDNA which have complementary sequences on the microarray. Perform washing steps and remove the unhybridized probe, and then scan the gel to look for the changes in the gene expression. In this manner, expression level of thousands of genes can be measured and analyzed by using DNA microarray platform simultaneously. After discussing some of the technologies used in genomics, let us now move on to transcriptomics. So, mRNA translation is a very intricate process which takes place on ribosomes. A study of all the mRNA molecules expressed by a particular cell type of an individual is known as transcriptomics. The transcriptomic analysis measures the genes that are being actively expressed or modulated at any given time and varies significantly with external environmental conditions, development conditions, or different type of environmental cues. Let us look at some of the definitions. A transcriptome is analysis of all species of transcript, including mRNAs, non-coding RNAs, and small RNAs. The transcript analysis is used to quantify the expression level changes of each transcript during development and under different conditions. The transcript analysis is also very important for understanding the functional elements of genome and also the molecular constituents of cell and tissue. There are various techniques which have been used for evaluating gene expression changes, such as Northern blotting, the more traditional method, and some of the newer methods involve quantitative real time polymerase chain reaction, qRT-PCR or qPCR, differential display method, serial analysis of gene expression or SAGE, and microarrays. Let us talk some of these techniques in some more detail. Reverse transcription PCR. RT-PCR is a variation of regular polymerase chain reaction or PCR that is used to generate multiple copies of DNA starting from a molecule of RNA. As you can see in the slide, the technique first requires the template RNA strand to be reverse transcribed into corresponding complementary cDNA, which is then amplified by using traditional PCR, and multiple copies can be generated. Reverse transcription PCR is used to generate multiple copies of DNA with RNA as the starting material. The template RNA molecule is first reverse transcribed into the corresponding cDNA by means of enzyme reverse transcriptase. This enzyme, which is commonly found in viruses, is capable of synthesizing DNA from an RNA template. Traditional PCR is then performed on cDNA obtained by addition of primers, which are allowed to anneal at 54 degrees centigrade. This is followed by addition of nucleotides and Taq polymerase, which performs elongation of template strand at 72 degrees centigrade. Second and subsequent rounds of PCR result in further amplification of the cDNA of interest. Strand separation is performed at 95 degrees centigrade, followed by primer annealing and elongation respectively. In this way, the mRNA transcript originally used is amplified in form of its corresponding cDNA, which can be studied further. A highly sensitive method which is employed to quantify the amount of RNA is known as real time polymerase chain reaction or quantitative PCR. This method requires very little, around 5 to 10 nanogram of RNA for detection of transcript changes. It quantifies a target DNA molecule as and when it is amplified by means of a suitable reporter dye that emits fluorescence every time a PCR cycle is performed. The double stranded DNA that needs to be amplified is heated to 95 degrees to bring about strand separation. Once the strands are separated, primers are added along with the probe DNA molecule, which have the quencher and the reporter molecule bound to its end. Once these have annealed to the template DNA strands at 54 degrees centigrade, Taq polymerase and nucleotides are added, and temperature is again increased to 72 degrees to carry out strand elongation. The Taq polymerase continues to elongate the DNA strand based on corresponding template DNA. When it reaches the bound probe molecule, the 5' to 3' exonuclease activity of Taq polymerase degrades the probe into its nucleotide fragments and continues to elongate the DNA strand. The released reporter dye thus gets separated from the quencher molecule during this process, and fluorescence emitted can be detected by using suitable detector. The increase in fluorescence in real time PCR is directly indicative of the amount of nucleotide being synthesized and is therefore a useful tool to measure the gene expression. As we have talked earlier, microarrays are a very strong platform for measurement of gene expression changes. The cDNA arrays measure many thousands of gene-specific mRNAs in single tissue sample simultaneously. As discussed earlier, the microarrays can be used for various applications including mRNA stability, identification of short poly-A phenotype and studying mRNA association with membranes or cellular organelles. The methodology is similar to what we discussed earlier. These large scale gene expression analysis has also proved to be useful as a validation strategy to validate the proteomics data and look at the changes at both protein level and correlate with the transcript level. A DNA microarray is printed with oligonucleotide sequences that will serve as probe molecules by providing complementary strands for binding cDNA of interest. Thousands of such spots, each containing as little as picomole quantities of DNA, can be printed on a single slide. Commonly used binding chemistries for printing include covalent attachment via epoxysilane, aminosilane, polyacrylamide, etc. The mRNA present in the control and test sample is extracted by addition of lysis buffer, which breaks open the cell. Centrifugation helps in pelleting down the cell debris, after which the mRNA present in the supernatant can be transferred into a fresh tube for further analysis. The extracted mRNA is then reverse transcribed into its corresponding cDNA by means of enzyme reverse transcriptase. This enzyme, which is also known as RNA dependent DNA polymerase, is capable of synthesizing DNA from a corresponding RNA template. The control and test cDNA samples obtained are then labeled with Cy5 and Cy3 dyes respectively. Since the two dyes can be excited at different wavelengths, the differentially labeled samples are then mixed together for further analysis. The mixed cDNA sample that is differentially labeled by means of two cyanine dyes is then incubated with printed DNA microarray slide. It allows hybridization to occur between the probe oligonucleotides on the array surface and the labeled cDNA samples of interest. The microarrays is washed to remove any unbound cDNA molecule and then is scanned at suitable wavelength by means of a microarray scanner. A new advancement in transcriptomic technology is RNA sequencing. RNA-seq has been used for transcriptome analysis by sequencing cDNA through next generation sequencing. This methodology was initially utilized for identification of transcriptional map of yeast. In RNA sequencing, the long RNAs are converted into library of cDNA fragments, as you can see here, through RNA or DNA fragmentation, and to each DNA fragment or cDNA fragment, sequencing adapters are added. From each cDNA, the short sequence is obtained and resulting sequence reads such as exonic, junction and poly-A end reads are aligned with reference genome sequence. So, the sequence reads exonic read, junction read and poly A N reads can be used to generate base resolution expression profile, as you can see here, plotted with nucleotide position on x axis and RNA expression level on y axis. The RNA sequence has dynamic range greater than 8,000-fold to quantify gene expression changes. Therefore, the RNA sequencing method is very useful for discovering new transcript, identifying mutations, deletions and insertions, as well as splicing alternatives. After studying genomics and transcriptomics, let us now move on to why do we need to study proteomics. So, after completion of human genome sequence, we identified around 30,000 genes, which were much less than expected number of 100,000 genes. The lower number of genes were surpassed by the estimated number of proteins, which is in millions. Therefore, studying large scale study of protein structure and function requires a thorough understanding of protein composition as well as information at various structural level by employing different type of proteomics tool. So, let us look at some of the definitions. So, proteome is entire complement of proteins expressed by genome of an organism under defined conditions, and the study of entire compendium of proteins encoded by a genome is known as proteomics. So, let us discuss why do we need to study proteomics and how it compares with genomics. So, there are various reasons if you compare genomics versus proteomics to look at the advantages and disadvantages of each method. The genome represents only the starting point towards understanding the complexity of biological functions. The proteins provide a much more meaningful insight into the complex biological system and the mysteries associated with it to understand the biological processes. So, unlike the genomic DNA, proteins do not contain intervening sequences introns and are directly indicative of cellular functions. Alternative splicing, it is a process by which exons or coding sequence of pre-mRNA produced by the transcription of a gene are combined in different methods during RNA splicing. The resulting mature mRNA gives rise to different protein products, as you can see in the slide, by the process of translation. Most of these are isoforms of one another. So, in this way a single gene can give rise to multiple protein forms. Another level of information which is only obtained at the proteomics level is post-translational modification. Many proteins undergo PTMs at some of their amino acid residues after the synthesis process. These modifications include hydroxylation, methylation, alkylation, phosphorylation, glycosylation, etc., which are some of the most commonly modified PTMs observed, but there are many other forms of post-translational modification which also occur in proteins. Very briefly, let us touch upon some of the techniques which are employed in proteomics. So, different type of proteomic technologies which employ gels can be grouped under the gel based proteomics. The protein within a cell are commonly analyzed using LDS page and two-dimensional gel electrophoresis. Separation in LDS page occurs almost exclusively on the basis of molecular weight. since all the proteins have similar charge to mass ratio and shape after they have bound the SDs. In 2DE, the complex mixtures can be resolved first by isoelectric focusing, followed by the based on size in polyacrylamide gel. Some of the limitation of two-dimensional electrophoresis were overcome by using two-dimensional difference gel electrophoresis or DIGE method. The 2DE or DIGE in combination with mass spectrometry has been the standard technique for proteomic analysis. Mass spectrometry, it is another technique for protein identification and analysis by production of charged molecular species in vacuum and its separation by magnetic and electric field based on m/z ratio. The different components of mass spectrometry: ionization source, it is one of the major components of any mass spec instrument which fragments the sample into ionic forms for further detection. Multi and electrospray ionization ESI are commonly used for protein sample analysis. Mass analyzer, another important component, resolves ions which are produced by the ionization source on the basis of m/z ratio. The various characteristics such as resolving power, accuracy, mass range and speed which determine the efficiency of these mass analyzers. The commonly used mass analyzers include quadrupole, time of flight, ion trap, orbitrap, etc. The mass spectrometry has become the method of choice for analysis of complex protein samples in proteomic study due to its ability to identify as well as quantify thousands of proteins. Protein microarrays, these are miniaturized arrays which are normally made on glass slide as well as other substrates onto which the small quantities of thousands of proteins are immobilized and analyzed. Protein microarrays can be generated either by traditional cell based method where proteins can be purified and then immobilized, or cell free methods where proteins can be produced by using in vitro transcription and translation system. These protein arrays can be used for various applications including protein-protein, protein DNA as well as measurement of protein activity and other biological applications. Very briefly, let us look at the label free detection techniques. Includes surface plasmon resonance or SPR. The label free detection methods monitor inherent properties of the molecule itself and overcome many limitations of traditional label based methodology. The various techniques such as SPR, SPR imaging, ellipsometry, interferometry, etc., which are commonly used for label free detection system. Now, shown here in the slide is surface plasmon resonance method, which detects any change in the refractive index of the medium of the material interface between the metal surface and ambient medium. So, in summary, today we talked about different techniques which are employed in genomics, transcriptomics, and we looked at why to study the proteomics. So, as we know, the biological systems are very complex to understand. The genomics, transcriptomics and proteomics can be used in various permutation and combination to address different biological questions and obtain information at different levels. An ideal approach would involve the systems biology based investigation, which we will discuss in the next class. Thank you.